Hello, everyone. I'm Camille Kotton, and I have the pleasure today to speak with Dr. Matt Yousefzadeh. Matt, could you tell us a little more about yourself?
Yes, I'm Matt Yousefzadeh. I'm an assistant professor at Columbia University Medical Center in the Columbia Center for Translational Immunology and the Birch Lodge Center for Human Longevity. So my lab works on aging, is particularly in the immune system and how that can drive systemic aging. And we have a special interest into transplant biology by getting to work with transplant biologists here and previously when I was a postdoctoral fellow.
So, Matt, tell me, how should transplant clinicians think about aging biology as a driver of graft dysfunction?
So I study senescent cells, which are particular cells that accumulate not only with chronological age, but biological age, and they can drive systemic aging through the increased inflammation and issues that occur with tissue dysfunction and impaired tissue regeneration. And it's even been shown that transplantation of senescent cells, whether the senescent cells themselves or in organs, into young recipients can actually cause secondary senescence and inflammation. And typically, this can occur is by the senescent cells secreting soluble inflammatory factors in what's known as the senescence-associated secretory phenotype that can not only cause tissue dysfunction, but could potentially aberrantly draw in inappropriate immune responses.
Thank you. That's really interesting. So, what do you think are the most important mechanisms linking immune aging to organ aging?
Great question. So we focus a lot on immunosenescence, which is this holistic aging of the immune system that occurs progressively, and it includes immune senescence, but a lot of other factors, such as loss of certain populations, gaining of others, changing in functionality, lymphoidal atrophy. So there's many features of it. And so what we can see is that not only are the immune cell types changing, not only by number, but by functionality. And globally, what we really see is that there's a loss of protective functions of the immune system, including loss of immune surveillance. So this could be against cancer cells or senescent cells, as well as the adoption of pathogenic phenotypes where adaptive immune cells can take on innate-like phenotypes, or even innate immune cells become hyperinflammatory. And this adoption of pathogenic features can sort of drive not only immune aging, but also systemic aging. And we think this also has great implications for transplant biology.
Thank you. That's really interesting. So, I have some further questions. One of them is, what are the most promising biomarkers or readouts to quantify immune aging and risk in transplantation?
Really good question. So currently in the field of geroscience, when we look at senescent cell burden, one of the most common assays that's used and has been used for over a decade is quantification of a senescence marker, P16, a cyclin-dependent kinase inhibitor gene, in peripheral blood T cells. So typically we isolate CD3 positive T cells and then quantify P16 or P21, another senescence marker in there. And this has been shown to not only increase with chronological age, but also biological age. And recently, we also showed that P16 expression in peripheral blood T cells correlated with baseline frailty and length of stay in liver transplant recipients. Furthermore, it may be more clinically relevant is the measurement of the senescence-associated secretory phenotype molecules. One of them in particular being GDF15, which has been shown to increase with biological aging, but also is recently shown to be predictive of risk of death even in healthy individuals as well. So we think these are really useful and translatable biomarkers that could be applied to the transplant field.
Excellent. So, there's probably something which many clinicians are wondering about. Where do the current clinical models fall short when it comes to accounting for biological age?
Also, a really good question. This is something that the aging field sort of grapples with as well. And so, what we've been using now are proxy biomarkers for biological age, and not just chronological age, the age that's on your driver's license. Is one thing that's grown recently over the past decade is the use of epigenetic clocks that are based upon measuring DNA methylation. And there's multiple generations. Some were trained on chronological age, then second generation clocks were trained on biological age or time to mortality. Third generation clocks currently look at your velocity or speed of age, and fourth generation clocks currently look at causal factors or adaptive factors with age. Luckily, all these generations of clocks use the same underlying data, so they could all be applied to looking at assessing age of the organ or recipient age at the time of transplant and down the road later on.
So I imagine at TTS 26, you'll have transplant professionals sitting in the audience, and one of the most important questions they'll ask is, what is the most realistic near-term opportunity for senotherapy to impact transplant patients?
That's something that we think a lot about too. And so one of the things we look at are what are not just cellular senescence, but also the hallmarks of aging. Which hallmarks are being perturbed in the transplant recipients? And knowledge of this can help us to build better combinations of drugs that we could use to alleviate some of the after effects of transplants and sort of the conditioning regimens that occur that can create iatrogenic genotoxicity and subsequent cellular senescence. So one thing we're particularly interested in, especially for senotherapies, are senotherapies typically target pro-survival pathways that are upregulated by senescent cells. And so we want to better understand in the conditions of transplant biology, which of the senescent cell anti-apoptotic pathways or SCAPs are upregulated so that we can better target them. And we think that will create more effective combinations.
Excellent. So once you're done with your talk at TTS 2026 and you're ready to absorb some knowledge yourself, what types of papers or presentations are you most hoping to see at the meeting?
So I'm really interested in looking at the biomarker field or the biomarker papers to sort of see the biomarkers that are being used in transplant biology. How much is there overlap with what we currently use in the field of aging biology or geroscience? Furthermore, papers or abstracts regarding the immune system and its role in transplant outcomes as well, because since we focus on immunosenescence.
That sounds great. So I can see that you're relatively early in your career. And can you tell me about mentorship, how important it was for you, and who did you have to help mentor you in your career?
So mentorship was incredibly important for me. I was very fortunate to get mentored by a number of people, not just sort of the standard academic PhD mentors, because I was a PhD myself, but also by clinicians and what's sort of clinically relevant. I'm incredibly fortunate to be in the Center for Translational Immunology and get mentorship from Dr. Megan Sykes, as well as some of the other people in the xenotransplantation field, like Greg Novak.
That is wonderful to hear. Megan Sykes and David Sachs are both long-term colleagues and friends. You know, they were at Massachusetts General Hospital before the transition to Columbia, and they have both had the enormous honor of receiving the TTS Medawar Prize.
They're so wonderful. I can't say enough positive things. And Megan and I sort of have a collaborative project with aging and some of her animal models. And you know, David is just fabulous. He's not always around here, but when I do get to meet with him, it's just so much fun.
Matt, thank you so much for such a wonderful interview. I'm really excited to hear your talk at TTS 2026.
Thank you for having me.